The Lotka-Volterra equations, commonly referred to as the predator-prey equations, are two first-order nonlinear differential equations that are frequently used to describe the dynamics of biological systems in which two species interact as either prey or predator. The Lotka-Volterra model makes a number of assumptions about the environment and the evolution of the predator and prey populations that are not always true in nature. The population of prey always has access to plenty of food. The size of the prey population is the sole determinant of the predator population's food supply. Population growth is inversely correlated with population size. The environment does not shift in favor of one species during the process, and genetic adaptation has little effect. The appetite of predators is insatiable. The growth equations for the two species are as follows: if one species uses some of the other's resources, dtk. For species 2d, the population sizes of species 1 and 2 are n and n. T. Population has a specific equilibrium level or k competition factors with coefficients denote the impact of species 1 and 2 on each other, accordingly. The population sizes of species 1 and 2 are n and n, respectively, whereas their individual growth rates are r1 and r, respectively. 1. New species 2 has a 1k1 inhibitory effect on itself and a 1k1 inhibitory effect on the growth of species. Any one of the competing species' growth will halt once it reaches its carrying capacity, which is the sum of its own and the numbers of the other species. Thus, species 1 stops growing when n plus n equals k. Similarly, species 2 stops growing when n2 plus bn1 equals kq. The two-species system will be at equilibrium only when the stoppage of growth coincides. For the two species simultaneously, that is, when dt equals dn underscore dt equals 0. 2. N is equivalent to k when n1 is a straight line and depicts all possible number combinations at which species 1 will reach its maximum size. The arrow at x indicates that if n and n are mixed, n will drop. Similar to how it is represented by the arrow, n will rise at a mix-up at y. O tilde kq. K2 of species 1. O at e equals 4. For instance, if there are a lot of mice in a paddy field, foxes would consume numbers of species 1. Here 2.16 CPG line of Lotka-Volterra. The predator-prey model of Lotka-Volterra is a straight yet useful model. This model makes the assumption that the population of prey determines how many predators are there and increase in birth rate. For instance, if there are a lot of mice in a paddy field, foxes would consume a lot of mice and produce a lot of young. The foxes will not be able to reproduce if there are no more mice. In contrast, because the predator population affects the prey mortality rate, the amount of prey would rely on it. For instance, if there are few foxes, the majority of mouse pups would grow. The mice would survive longer, even the older ones. As a result, prey are limited by predators, and predators are limited by food. At any point along line C, the predator population exhibits neither growth nor reduction, dn predator dt equals O. The population of predators will drop at any point to the left as a result of scarcity of food. Similar graph can be made for the population of prey. At any point on line D, the population of prey will remain the same, dn prey dt equals O. EOPUL at ION. SIZ. OF. PRED at R. N. Population C. A. Predator population declines, increases due to lack of, plenty of food. 3. Putting the two diagrams, one can analyze the system's behavior and find that at point E, dn predator dt and dn prey dt are both equal. The system is currently in balance, with neither population increasing in size. One population would, however, be drawn away from this point in any natural system. The deep arrows forming a circle around E indicate the likely outcomes of the system. The population would rise in the quadrant indicated by A, and the system would migrate to the upper right quadrant. In this case, the tendency would be for the predator population to continue growing while the prey number would decrease. Once in the upper left quadrant, both populations would start to fall as the system moved there. Eventually, the system would go back to A. Prey F declines because predation. E. OF. PRED at OR. Population size of prey B. Figure 10.17a predator population B prey population. Social ecology. The definition of community ecology is quite semi of LSY and eco R log TY. O com timun hity isa tvir y old notion that dates back to Theophrastus's time, 370-250 BC. A community, often referred to as an ecological community, a biotic community, or biocenosis, is a collection of populations that coexist and interact in a particular location for a period of time. 4. The coexistence and interactions of numerous populations of trees, shrubs, herbs, grasses, animals, and microorganisms, or reflected in a forest community. In other words, it is the aspect of the ecosystem that is biological as opposed to abiotic. A community was once supposed to function as a single thing, leading to the interpretation that it was a superorganism. A different perspective, in contrast, saw a community as a collection of species, each of which has its own identity. Every community has physical borders. Communities may be separated by boundaries that are sharp. However, the intensity of sharpness varies, such as a barrier between a forest and a lake, or a forest and a grassland. Community. There is frequently an ecozone, or transitional area, between two communities, where species from both nearby communities can be found. has made the ecological communities rich in species diversity, contrasting environmental conditions at the boundaries or the edges supporting a high species richness. An area of land between two forest communities, for instance, will feature plants and animals that are typical of both forest communities. Characteristics of communities. Communities can have a variety of traits, including species diversity, organizational structure, trophic structure, composition, dominance, succession, and developmental history. The following is a discussion of each of these. Species diversity. Taxonomically diverse species constitute each community. The amount of distinct species in the community, including both common and unusual species, is referred to as species diversity. Contrast to physically or human-controlled ecosystems, species diversity is quite low in natural communities like tropical rainforests or coral reefs in oceans. The two elements of species diversity are species richness and species evenness. Species richness can be defined as the variety of species present and their relative abundance. In a sense, it relates to the proportion of species S to all species N. A metric used to determine how evenly distributed species are in terms of numbers is known as species evenness. It refers to the distribution of each species within a community. For instance, a community is evenly distributed if it contains 10 species with 10 or 9 individuals per species, as opposed to 10 species, in which one species has 90 individuals and the other 9 species include only 10. Different diversity indices, which are mathematical expressions based on data on species abundance, can be used to quantify species diversity. The variety of species can be counted separately as species richness, evenness, or overall diversity. The WF index of richness, designated by R in the formulas in box 1, calculates the diversity of species 1958. The evenness index designated by Hill as E can be used to assess the evenness of a species 1973. The Shannon-Weiner index, also known as Shannon's index or simply the index of diversity denoted by H, as given by Shannon and Weaver, 1963, and the index of dominance or X, also known as Simpson's index, as given by Simpson, 1949, can both be used to calculate the diversity of a species, see box 1. While the index of dominance has an inverse relationship with species diversity, Shannon's index has a direct correlation with it. The formulas are provided for computing various species diversity indexes. Richness index. Index of dominance A equals Zn. Index of diversity. Evenness index E equals, where S total number of all the species in equals the ith species. As per Margalef 1958. As per Simpson 1949. As per Shannon Weaver. As per Hill 19. Tilde, total number of individuals AR of individuals of. Figure 10.18 Important formulae. A community needs both resilience and resistance to stay stable. A community's resilience is its capacity to bounce back from a disturbance or eviction. Contrarily, resistance is a community's capacity to fend against disruption, any event that modifies a community's structure, or resist a community's capacity to fend against disruption, any event that modifies a community's structure, or relocation, shifting of the community to some other place. Fact that any change in one or a few species would be made up for by the other species is one of the reasons that species-rich complex communities have become stable. However, some other researchers have shown that a society becomes unstable as its complexity increases. Therefore, it is debatable whether a community's complexity results in stable or unstable circumstances. Ecologists have, however, been effective in establishing a link between competition and variety or stability. If there is intense competition, there will be little diversity, since only those species will survive that can adapt to the challenging environment and resist it. On the other side, if there is no competition and there is little overlap in the needs of the species, there will be no fighting over resources and more species will be able to cohabit. If the resources for the life support, food, air, water, space, and sunlight are few and the needs of the species overlap, the struggle will be fierce. Community organization and makeup. Every community has a unique structure and makeup. For example, the rainforest community in the Silent Valley will be distinct from the one in Arunachal Pradesh. The dominant growth form of a community, such as trees in forests or grasses in grasslands, is frequently used to describe the community's structure. The community's structural layout is determined by how the various growth forms are arranged. The physical configuration of the elements within a community is also crucial. For instance, in a forest, some plants may prefer the shade and only grow in the understory, while others like emergent trees are adapted to harsh sun exposure. However, certain plants may flourish in dry, stony locations, while others require superior soil moisture. Similar to this, in a pond environment, the soil-dependent plants will stay around the pond's boundaries, while the free-floating species would blanket the water surface. Zonation describes the way a community is spread out horizontally. Life forms. Christen C. Ronkier, a Danish botanist, proposed the concept of life forms, also known as Ronkier's life forms or botanical life forms. Ronkier says that it is crucial for a community to understand how a plant survives in challenging circumstances. He saw the criterion of persistent buds being protected from harmful conditions as a climatic adaptation of the plant. Ronkier's approach classified plants into different life forms depending on where their buds were during seasons with unfavorable conditions, too cold or too hot. Ronkier gave phanerophytes, chamophytes, hemicryptophytes, cryptophytes, and therophytes as his top 5 categories of life. 1. Sometimes known as plants with visible buds, i.e., phaneros, visible, phytes, plants. These are the plants whose buds are located on the tops of shoots, high up on the plant. These are either scale-free or completely nude. The prevalence of phanerophytes increases in tropical regions, while it decreases in temperate and polar regions. Phanerophytes are further split into 4 types based on the height of trees. A. Megaphanerophytes are trees that are taller than 30 meters. B. Mesophanerophytes are trees between 8 and 30 meters in height. C. Microphanerophytes are trees between 2 and 8 meters high. D. Shrubs that are taller than 25 centimeters but less than 2 meters are known as nanophanerophytes. 2. Camophytes. These are plants with buds that are either very close to the ground or reach a maximum height of 25 centimeters. In colder plants at higher altitudes or latitudes, such as temperate America, these plants can be found. The aerial parts of camophytes can perish throughout the growing season and conceal the buds. Fresh growth begins as soon as a favorable season arrives. 3. Hemicrypts. These are also found in cold climates where buds are shielded from the elements by remaining above ground but not deeply rooted. These include herbs that are annual or biannual, have a life cycle that lasts 1 year or 1 season and 2 years or 2 seasons, respectively. 4. Geophytes. These are also referred to as cryptophytes. The buds of these plants, which include bulbs and rhizomes, continue to be buried in the ground. These plants are primarily found in the world's dry regions. 5. Theraphytes. These are ephemeral or seasonal plants. When conditions are favorable, go through their entire life cycle swiftly, and when those conditions are not favorable, they persist as seeds. Are also classified epiphytes, or plants that grow on or are attached to other plants, as a distinct category of life form, in addition to these 5 main groups. He classified cryptophytes into 3 subtypes: halophytes, hydrophytes, and geophytes, which are aquatic plants that are immersed or floating in water and have subterranean or perenniting buds, plants in marshy swampy areas with high salt concentrations. Environmental succession. Due to variations in climatic and physiographic conditions and the actions of species and groups themselves, the environment is constantly changing over time. These influences cause noticeable changes in the community's dominance, which are eventually replaced by a new community in the same location. The process keeps going, and succeeding communities grow one after the other in the same area, until the last community once more stabilizes for a while. Ecological succession is the occurrence of generally distinct sequences of communities over time in the same region. With time and space, plant communities are always evolving. Communities might be experiencing these changes as a result of their own internal dynamics or shifting environmental situations. As a result, in a region, one community may displace another, which may then be replaced by another community. Ecological succession refers to the gradual change in a community's structure and makeup over time, leading to the establishment of a stable community. Some ecologists refer to ecological succession as ecosystem development. It is a systematic and predictable process. The final stable community that is created at the conclusion of succession lasts a long time and coexists peacefully with the surrounding environment. The communities that appear for a brief period of time and are replaced one after the other are referred to as seral communities or seral stages. This final stable community is known as the climax community. The first plant community to settle in a region is known as a pioneer community, and the plants that make this community are known as pioneers or primary colonizers. Succession processes. Primary-secondary succession. A succession is referred to as primary if it begins with the primary substrate, which is devoid of any living being or organic substances, and uninhabited environment. It is a slow-moving ecosystem that can exist in terrestrial or aquatic settings. In contrast, secondary succession begins from a substratum that has already been developed. In such locations, disturbances like rapid climate changes, fires, floods, or symbiotic interference cause the plant community that had previously inhabited the area to change. Secondary succession is the word used to describe the relatively quick succession that begins from such a substratum. Autogenic and allogenic succession. During the process of succession, the environment is changed by the mutual interactions of the living members of the community. The existing community is forced to be replaced as a result of these changes, making the environment unfavorable for it. Such modifications might, however, be beneficial for another community. Autogenic succession is the process wherein a community is replaced by factors that the community itself has generated. Allogenic succession is used to describe a succession in which the replacement of one community by another may be influenced by environmental causes rather than by living things. Heterotrophic and autotrophic successions. An autotrophic succession is one in which green plants or autotrophs predominate so that there is a constant flow of energy throughout the entire process. Contrarily, heterotrophic succession is characterized by the dominance of heterotrophs like bacteria, fungus, actinomycetes, or even animals and a persistent drop in the energy flow. Additionally, it might be one of the following varieties depending on the environment, mainly the moisture conditions, at the start of succession. Hydrarch or hydrozer. If a succession starts from a water body, it is referred to as a hydrarch. Or hydro. A lake, pond, stream, bog, or even a swampy area could represent the water body. Xerozer or xerarch. In this instance, the succession starts from dry, low-moisture environments like a desert, sand dunes, and boulders. Lithozer is then given to a succession that specifically begins in rocky terrain, whereas P.S. Amozer is the name given to a succession that specifically begins in sand dunes. Mesozer or mesarch. The succession is referred to as mesozer or mesarch if it begins in an area with suitable temperature and moisture conditions. Halozer, succession that develops in salty environments, or in places where the substratum has a high concentration of salt. Community structure. The phrase, community structure, refers to a broad category of phenomena that emerge from the creation of ecological communities. Community structure is a term used frequently in ecology to describe the boundaries that species interactions place on which different species can coexist. These restrictions on coexistence are known as assembly rules, and they can be used to limit the coexistence of both individual species and groups of species, such as the guilds described below. The part that competition has played in community structuring has been of special concern to ecologists. The following section will go into further detail about this. The term, community structure, is sometimes used to refer to how species are physically arranged within a community, such as how trees, bushes, and ground-dwelling herbaceous, non-woody plants are arranged vertically in a forest. Energy flows in food webs. An introduction: we are all aware that the largest and toughest predators, such as tigers and great white sharks, are relatively uncommon on Earth in comparison to other species. Do you realize why? In early days of the field of animal ecology, a scientist by the name of Charles Elton took the common knowledge that big fish eat little fish, and turned it into an organizing principle that is still in use today, that plants and animals are organized into trophic or feeding chains and food webs of interaction. He also established the concept of a pyramid of creature numbers, where, for instance, there are many plants at the base of the food web, fewer herbivores that graze on those plants, and even fewer predators that devour the herbivores. Thus, Elton observed the world around and concluded that large, fierce creatures were uncommon, but he did not know why. The mechanism, or reason for the scarcity of top predators in food webs eluded him. We will discover this process and answer the posed question by studying the nature of the energy flow in ecosystems. As energy travels through ecosystems, it is used and lost as heat. While new energy is continuously provided to the Earth in the form of solar radiation. An open system in terms of energy, but a closed system in terms of elements such as nutrients that are continuously recirculated within and across ecosystems. Energy and materials are vital to the structure, function, and composition of an ecosystem. The fundamental concepts of nutrition cycles have previously been introduced. This section will focus on energy. In terms of carbon cycling, note that materials and energy can be converted. For instance, we know how many a unit of energy, a gram of carbon compounds such as lipids or carbs have. Autotrophs versus heterotrophs. As a brief review, we acknowledge that certain species synthesize organic compounds from inorganic substrates and store biochemical energy in the process. These are referred to as autotrophs, which means self-feeding. Autotrophs are also known as primary producers. Plants, some protists, and some bacteria are able to produce complex organic molecules from simple inorganic substances, water, colorado, and nutrients. This is often accomplished by photosynthesis, which, as its name suggests, requires light. We should also discuss the pathway known as chemosynthesis for completeness. Certain producer organisms, primarily specialized bacteria, can transform inorganic resources into organic chemicals in the absence of sunlight. There are numerous chemosynthetic bacteria in marine and freshwater ecosystems, especially those rich in sulfur or hydrogen sulfide gas. Chemosynthetic creatures, including chlorophyll-containing plants and other species capable of photosynthesis are autotrophs. Numerous creatures can only acquire energy from other organisms. They are known as heterotrophs. They include consumers of all forms of organisms, including plants, animals, bacteria, and even decomposing tissue. Heterotrophs are also known as consumers. The primary production. The phrase, production, often refers to the development of new organic matter. When a crop grows, photosynthesis creates new organic matter by converting light energy into energy stored in chemical bonds within plant tissue. This energy fuels the plant's metabolic mechanism. Over time, the plant synthesizes new substances and structures, the cells divide, and the plant expands in size. Plants require light, carbon dioxide, water, and nutrients, and through photosynthesis, they make reduced carbon compounds and oxygen. Is the rate at which photosynthesis occurs or the rate at which an individual plant grows in size. Primary production is the focus, the synthesis and storage of organic molecules during the growth and reproduction of photosynthetic organisms. The fundamental concept is that new chemical molecules and plant tissues are generated. Primary production results in the addition of new plant biomass to the ecosystem through time. Consumers obtain their energy either directly, herbivores, certain detritivores, or indirectly, from secondary producers, from primary producers, predators, other detritivores. Is there a limit to the maximum primary production? Yes is the quick answer. Let's take a quick look at the amount of energy captured by autotrophs and the efficiency of photosynthesis. Recall the strength of solar radiation reaching the Earth's surface varies in part on the location. The equator receives the most energy, while the intensity diminishes as we approach the poles. As we learned in the lesson on ecosystems, these differences have significant effects on climate and result in the observed biome distribution patterns. In addition, we know that just a small portion of the sun's radiation is utilized by the photosynthetic response in plants at gross primary production, GK. Respiration, R, is the amount of CO, V. Respiration can be further divided into compo. RP equals respiration by plants. R equals respiration by heterotrophs. Ri equals respiration by decomposers, the microbes. The surface of the Earth. About half of the total solar radiation striking the Earth's outer atmosphere is reflected back to space by ice, snow, oceans, or deserts, or absorbed by gases in the atmosphere. For instance, the atmosphere's ozone gas layer absorbs nearly all ultraviolet rays, which accounts for about 9% of the sun's radiation. Only about half of the rays that reach the Earth's surface is in the wavelength range that plants can use for photosynthesis, 400–700 nanometers. This is known as photosynthetically active radiation or PAR. Plants greatly absorb light of blue and red wavelengths, as well as light in the far infrared range, thus their green hue, the result of reflection of green wavelengths, and also reflect light in the near infrared region. Even if the correct wavelength is used, photosynthesis does not convert all of the light energy into carbon. Some of light does not reach chloroplasts, where photosynthesis reactions take place, and much of the energy from light that is converted by photosynthesis to carbon compounds is used to maintain the plant's biochemical machinery. This loss is commonly referred to as respiration, although it also includes thermodynamic losses. Therefore, plants do not utilize all of the potentially available light energy. Plant in photosynthesis. System from metabolic activity. Source of the CO. Net primary production, NPP, is the net amount of primary production after the costs of plant respiration are include. Therefore, NPP equals GPPR. Net ecosystem production, NEP, is the net amount of primary production after the costs of respiration by plants, heterotrophs, and decomposers are all included. Therefore, NEP equals GPP, RP plus RI, plus RI. Here 10.19 Key formulas. Evaluation of primary production. Due to the simplicity of the basic technique, the primary production of the world's oceans has been meticulously mapped, and numerous freshwater lakes have also been explored. Half of a series of miniature glass bottles with stoppers are wrapped with a substance such as tinfoil to prevent light from penetrating. These are respectively known as the bright and dark bottles. The bottles are filled with water from a certain location and depth. This water contains the aquatic ecosystem's microscopic plants and creatures. The bottles are sealed with stoppers to prevent the interchange of gases or organisms with the surrounding water and are then suspended at the same depth for a few hours. In some bottles, Colorado is consumed and O is created, and the change in either of these gases over time can be measured. Using chemical titration, for instance, the amount of oxygen dissolved in water can be easily quantified. Before suspending the bottles, the initial O concentration is measured and expressed in milligrams of oxygen per liter of water, milligram per liter. After a scheduled incubation period, the final value is measured in both the light and dark bottles. What actions are occurring in each bottle that may affect the initial O and Colorado concentrations? The following equations describe them. Light bottle, photosynthesis or gross primary production, GPP, and respiration occur in the light bottle R. As we observed previously, the difference between these two processes is net primary production equals NPP equals GPPR, dark bottle. There is no photosynthesis and solely respiration in the dark bottle. In this example, there may also be some consumer respiration in both bottles, unless a net was employed to remove heterotrophs. Consider the following simple illustration. Demonstrates how we account for changes in the original oxygen concentrations in water that happened during incubation. We will assume that our incubation period lasted 1 hour. Oxygen concentrations measured. First is 8 mg O, L, second bottle equals 10 mg 0, L, third bottle equals 5 mg O, L. Due to photosynthesis, the amount of oxygen in the light bottle rose relative to the starting amount, whereas the amount of oxygen in the dark bottle decreased. With this information, we can calculate our system's respiration, NPP, and GPP. Initial light equals 10 8 equals 2 mg per liter H equals GPPR equals NPP. 8 5 equals 3 mg per liter H equals respiration, light dark equals 10 5 equals 5 mg per liter per hour equals NPP plus R equals GPP. Thus, we have a measurement of our systems net and gross primary output, as well as its respiration. When primary output is very low, the oxygen approach has its limitations. C14CO, a radioactive form of carbon, can be used to monitor carbon intake and fixation in certain instances. In order to convert the results between the oxygen and carbon approaches, multiply the oxygen values by 0.37. The factor comes from differences in atomic mass. Biological diversity is defined as the variety of all life forms, including various plants, animals, and microorganisms, as well as the genes and ecosystems they generate. The definition is not just species variety but also genetic diversity of individual organisms, habitat diversity, natural community diversity, and ecological process diversity throughout the biosphere. Food chains. IT is an organismal chain through which nutrients and energy pass, from producer to the main consumer, then to the secondary consumer, followed by tertiary consumer to quaternary consumer to detritus feeder. It is the general order of the food chain. Herbivores are the main consumers. Everyone else is either a meat eater, carnivore, or both omnivores. The food chain in a field ecosystem might look like this: grass, grasshopper, mouse, snake, hawk. Detritus versus grazing food chains. Detritus food chains are based on detritivores which break down detritus and wastes, whereas grazing food chains are based on producers' photosynthesis. Wild detritus chains, grass, greater than rabbits, are significant in forest litter. Grazing food chains are significant in marine and aquatic environments. Simple inorganic. Cats ST materials less than. Wolves, greater than tigers. Decomposers bacteria, fungi of decay. Figure 10.20 Food chain. Food web. Of course, the real world is more intricate than a straightforward food chain. Food webs are made up of connected food chains rather than discrete food chains. The food chain and ecosystem are more stable the more alternate avenues there are for energy to pass through. Since there are producers at the bottom of the food webs you can see, which eat herbivores subsequently graze on, these are grazing food chains. Although grazing food networks are significant, detritus-based food chains exceed them in nature. The decomposers at the bottom of the food chain and detritus-based food chains support the predators that consume them. In many ecosystems, debris contributes a greater portion of an animal's weight or biomass than living producers do. Diatoms, Desmic. W. Nessium Insic. The energy flow brought on by solar radiation's influences to occurrences that are closely related to one another and have their roots in the Earth's surface. It initially allows plant cells to come in contact with dissolved minerals, lakes, oceans, or saline soil solutions. This naturally results in cellular assimilation. The energy input required by chlorophyll to create organic matter, and subsequently living matter, is then ensured. 1.2% of the sunlight that reaches plants is actually absorbed, and roughly 1/3 of that energy is converted into chemical molecules that other organisms might consume. The plant's biomass is made of these organic components. About 10% of eaten biomass can be converted into biomass by organisms at higher trophic levels. A herbivore may therefore gain 1 kilogram of weight after consuming 10 kilograms of plant material. MGFC N productivity equals M. Primary manufacturing. Primary production describes the pace at which a specific amount of organic matter is created from mineral matter and energy in a particular amount of time. It is measured as mass of carbon assimilated per interval of time. Autotrophs, also known as primary producers, are the first trophic level or link in what is often referred to as the food chain and are responsible for primary production. Plants produce their own matter through photosynthesis, and consequently from photons, are the most well-known autotrophs. Additionally, there are autotrophic microbes that use chemical reactions to provide energy for production. These are referred to as chemosynthetic creatures or chemoautotrophs. Efficiency. The majority of authors use the wrong definition of productivity, which is the amount of biomass generated per unit of surface area over a specific period of time, or annual net primary production. Actually, the ratio of production to habitat biomass at any particular time serves as a measure of productivity. Efficiency equals PP. It is represented with a unit that is opposite to time T1. Productivity is represented as follows for plankton in an aquatic environment where the primary production is calculated in milligrams of carbon assimilated per cubic meter of water per hour. The rate of biomass renewal, or turnover, is its inverse. Renewal rate equals BP. The plankton renewal rate in the above cases, MGFC per meter 3 per hour is the rate of renewal. Plankton are extremely productive. Cells can divide in a day when the conditions are right. Therefore, their renewal rate is 1 day every year. For terrestrial plants, this turnover varies by organ and plant and is substantially lower. Thus, for leaves, it is 1.1 each year, while for wood, it is 0.02 per year. Transfer of energy and matter. For a trophic level, energy efficiency is defined as the ratio of energy released to energy consumed, or, more specifically, as the ratio of energy fixed to energy received. Efficiency of consumption. The consumption efficiency is the ratio of the energy that a consumer integrates to energy that is present in the field at hand. This will be the portion of appraised energy that the predator can efficiently incorporate. Not all of the consumer accessible net production is used. The entire net productivity of a plant is not consumed by a herbivore. For instance, it bursts sensitive shoots and discards roots and woody branches. The bones and shells that a carnivore leaves behind are filled with energy. The efficiency of consumption varies greatly depending on the ecology. Efficiency of assimilation. As food travels through the digestive tract, it undergoes both mechanical and chemical digestion processes. Only a portion of the food, the nutrients, enters the bloodstream through the intestinal mucus. The remainder is not absorbed and is expelled with the feces. The energy assimilated A by nutrients and energy E assimilation equals AL constitute the assimilation efficiency. Depending on the types of consumer organisms, it varies a lot. The phytoplankton environment when there are significant inputs of mineral components like nitrogen and phosphorus, and this growth is referred to as bloom, or flowering of water. Under these circumstances, the integrating filtering zooplankton consumes far more organic matter than is necessary for its metabolism. A significant portion of the food consumed is released as feces, which settle in the deeper zones and are eaten by other creatures. Since these feces contains phytoplanktonic cells, which are not killed by the zooplankton's digestive enzymes, the growth is aided by nitrogen and phosphorus released during the enzymatic activities. The biomass of the consumer does not increase as a result of anything that is ingested. Because it is needed to meet the metabolic requirements of respiration, excretion, or home other meat, a sizable portion of the energy taken in is not fixed. This allows us to define another type of yield, the net production efficiency, which corresponds to the ratio of fixed energy, i.e., net energy N over assimilated energy AE net production is equal to O. Ecological efficiency. Because it gauges a trophic relations yield, ecological efficiency is the most pertinent to our discussion. It is also known as ecological growth yield, feed efficiency, or ecological yield. It is the sole ratio that captures the energy losses incurred as feed levels are changed. As a result, it has a considerable benefit when trying to comprehend how biocenosis work. It is described as the proportion of energy ingested I found in the feed consumed to energy fixed N in the net production of a consumer. E environmental equals Ni. Ecological efficiency, for instance, is the ratio between a predator's net production and the net production of its prey. It has to do with the ratio of energy values, not of matter weights. The various degrees of diversity. The term diversity is most frequently used to describe species diversity, which is a measurement of number of species and their relative abundances within a community. Despite the fact that the species is the taxonomic level that is most frequently employed, research at biogeographical scales also assesses the diversity of genera, generic diversity, or families to compare regions. A similar metric known as genetic diversity assesses a variety of genotypes found within a species or population. Through the selection of crop plant types in domestic animals, humans have long been aware of the concept of genetic diversity. The term landscape diversity has only lately been created to describe a variety of communities or vegetation kinds that exist at the size of the landscape. Genetic diversity. Genotype variety within a population or species is referred to as genetic diversity. The phrase might refer to a specific quality, e.g., the color of corn grains, resistance to disease. In a broader sense, it refers to the entire degree of diversity for all qualities. Subspecies are populations that seem to be genetically distinct from other populations of the same species, a geographically recognizable population. Races and varieties are terms for human-modified genotypes with reference to animals. In fact, the idea has long been used in agriculture and animal breeding, where breeders have created new kinds by exploiting the natural genetic varieties of wild parents and relatives of cultivated species. Textulating genetic diversity study of morphology. If a trait has a simple genetic determinism, i.e., it is determined by one or a few genes, it is feasible to analyze genetic variety by merely counting the frequency of the various phenotypes present in the population. Polymorphism is the presence of two or more phenotypes in a population. A variety of distinct protein forms within a population can be determined using the chemical technique of electrophoresis. Measuring the variation in these proteins indirectly gauges the genetic diversity because the various forms are a direct result of the variation in the underlying genetic code. Sequencing techniques. Molecular biological technologies have recently been developed to delve directly into the DNA composition. Genetic diversity may be measured because of individual variation in the DNA make. What makes genetic variety crucial? Genetic variety is a key factor in determining how adaptable it is to environmental change, unintentional population declines, and attacks from adversaries like illness. Genetic diversity can be thought of as a species, life insurance, against these threats. Without such variation, natural selection cannot work. And extinction may ensue from environmental changes if those in the population who can adapt to them are not present. Diversity of species. Indices of diversity. If you want to compare communities or regions, using a standard measure of diversity is needed. A diversity index is a straightforward number that may be used to summarize the data in a list of species and their abundances. There are several of these indices. The total number of species in a community is the simplest index to use. This is typically denoted by the symbol S and is known as species richness or sometimes N. The drawback of species richness is that species abundances are not taken into account. There are two widely used indices that consider species abundances. D equals 1 D, Simpson Diversity Index. H equals P, in P, Shannon Diversity Index, where pi is each species' relative abundance. It will allow you to observe the results of including abundance. It should be noted that these indices take into account both variations in abundance and richness. In order to determine how equally the species are distributed, we may only wish to take into account the relative abundances of the species, rather than the species richness. In order to do this, D or H can be expressed as a percentage of the value that would be anticipated if all species were present in equal abundance. Simply divide D by the species richness and H by the logarithm of S to accomplish this. In example, observe that community 1 is significantly more diversified, D equals 5.0 cf. 1.06, H equals 1.61 cf. 0.21, while having the same number of species, S equals 5, as community. Also, observe that communities 1 and 3 have the same level of evenness, D, S equals 1, H, ln S equals 1, while having differing numbers of species. The indicators at several spatial scales, diverse geographical scales, are acknowledged when diversity is taken into account. The diversity found in a community sample is referred to as alpha diversity. The diversity among community samples is referred to as beta diversity. So we are aware that various species can frequently be found in various settings. Explaining beta diversity normally presents little difficulty. In contrast, it can be quite challenging to explain alpha diversity. Hutchinson, 1061, posed the paradox of the plankton, which is one of the most well-known discussions of alpha diversity, in which he questioned how so many phytoplankton species could persist when they all share the same environment and fight for the same basic resources, e.g., light and nutrients. Gamma diversity, or the fusion of alpha and beta diversity, is diversity at the regional level. The diversity of species fluctuates over time as well. In terms related to biological diversity, totality of the species, genes, and ecosystems in a given area. Genetic diversity, it refers to gene variety within a species. Ecological diversity, variation in a given area's habitats, community types, and abiotic environment. In situ conservation, keeping an organism in its natural environment is known as in situ conservation. Ex situ conservation, it includes cryopreservation, tissue culture, and the preservation of organisms in botanical gardens, zoological parks, and sanctuaries. Hotspot, priority conservation regions that are both highly species-rich and imperiled by extinction. Exotic or alien species, these are animals or plants that have been brought into an ecosystem from elsewhere. Biosphere reserves, these are safeguarded coastal or inland environments with distinctive biodiversity. Sacred forests, sacred groves, these are forest swaths that tribal communities guard because of their religious convictions. Biomaterial is stored at extremely low temperatures using cryopreservation. Red List Guide, the IUCN issued the book, which includes a list of species that are recognized to be endangered. The Program on Man and Biodiversity, MAB, the World Wildlife Fund for the Environment, Indian Board for Wildlife, IBWL. Biological Program, International Union for the Conservation of Nature and Natural Resources, IUCN, United Nations Development Program, or UNDP. Biogeochemical cycles. Through the numerous transfers between trophic levels, energy enters ecosystems as sunlight or inorganic compounds for chemoautotrophs and exits as heat. The material that makes living things is preserved and recycled. Nevertheless, carbon, nitrogen, hydrogen, oxygen, phosphorus, and sulfur are the 6 most frequent elements linked to organic molecules. They can persist for extended periods of time in the atmosphere, on land, in water, or below the surface of the Earth in a variety of chemical forms. This material recycling is influenced by a number of geologic processes, including weathering, erosion, water drainage, and the subduction of continental plates. Biogeochemical cycle is the term used to describe the recycling of inorganic matter between living organisms and their environment. Geology and chemistry play significant roles in the study of this process. Hydrogen and oxygen, both of which are necessary for all biological processes, are found in water. The hydrosphere is the region of the Earth where water is stored and moved. This water can be in the form of liquid on the surface and below it, frozen rivers, lakes, seas, groundwater, polar ice caps, and glaciers, or it might be in the form of water vapor in the atmosphere. All organic macromolecules contain carbon, which is also a crucial component of fossil fuels. Our nucleic acids and proteins include a significant amount of nitrogen, which is also essential for human agriculture. One of the components of artificial fertilizers used in agriculture and their accompanying environmental effects on our surface water is phosphorus, which, along with nitrogen, is a major component of nucleic acid. When fossil fuels like coal are burned, sulfur is released into the environment. Sulfur is essential for the 3D folding of proteins, such as in disulfide binding. These elements cycles are intertwined. For instance, leaching of nitrogen and phosphate into rivers, lakes, and seas depends on the flow of water. In addition, the ocean itself serves as a significant carbon sink. Mineral nutrients are thus cycled, either quickly or slowly, between the biotic and abiotic worlds, from one living thing to the next. The hydrologic cycle of water. The foundation of all biological activities is water. Human cells contain more than 70% water, and the human body is more than half water. So, to survive, the majority of terrestrial animals require access to fresh water. 97.5% of the water reserves on Earth are saline water, which cannot be used for drinking. About 99% of the remaining water is frozen or kept underground as water. Therefore, only 1% of fresh water may be conveniently accessed from lakes and rivers. The tiny amount of fresh surface water supply is crucial for the survival of several living creatures, including plants, animals, and fungus, and its absence can have a significant impact on ecosystem dynamics. Humans have created technology to improve the availability of water, such as drilling wells to gather groundwater, collecting rainwater, and desalinating ocean water to produce potable water. The availability of fresh water is still a significant problem in contemporary times, despite the fact that this pursuit of drinkable water has been ongoing throughout human history. The hydrologic cycle, water cycle. Evaporation. In ice, underground, and in the ocean. The cycle of carbon. In living. Condensation, precipitation. In ice, underground, and in the oceans. The following are some of these procedures: evaporation, sublimation. The habitats of ecosystems, some of which climate and by extension. The habitats of The dynamics of ecosystems depend a lot on the water cycle. Climate and by extension. The habitats of ecosystems, some of which are found in remote regions of the Earth, are significantly influenced by water. The majority of the water on Earth is permanently trapped in ice, underground, and in the oceans. The following are some of these procedures: evaporation, sublimation, condensation, precipitation, movement of subsurface water, snowmelt, and surface runoff, streamflow. The cycle of carbon. In living things, carbon is the second most common element. Since carbon is a component of every organic molecule, living things place a high value on its function in the formation of macromolecules. Those originating from fossilized organisms, primarily plants, which humans use as fuel, carbon compounds have a particularly high energy content. There are now more nations than ever before that consume enormous amounts of fossil fuels. Global demand for the finite supply of fossil fuels on Earth has increased since the start of the Industrial Revolution, which has led to an increase in the amount of carbon dioxide in our atmosphere. This rise in carbon dioxide is a serious environmental concern across the globe, since it has been linked to climate change and other disruptions of the Earth's ecosystems. As a result, the carbon footprint is determined by the amount of carbon dioxide produced in the consumption of fossil fuels by various nations. It is easiest to study the carbon cycle as two interconnected subcycles. One that deals with the quick exchange of carbon among living things, and the other that deals with the slow cycling of carbon through geologic processes. Carbon moves through the air, water, and soil in a complex manner that frequently happens much more slowly geologically than it does between living things, in what are referred to as carbon reservoirs, which include the atmosphere, liquid water bodies, primarily oceans, ocean sediment, soil, terrestrial deposits including fossil fuels, and the Earth's interior, where carbon is retained for protracted periods. As previously mentioned, the atmosphere serves as a significant carbon dioxide storehouse. Carbon dioxide is crucial for photosynthesis. The oceanic carbon reservoir has a significant impact on the amount of carbon dioxide in the atmosphere. The amount of carbon found in each place is influenced by the circulation of carbon between the atmosphere and water reservoirs, and each has a reciprocal impact on the other. When atmospheric carbon dioxide, CO, is dissolved in water, it reacts with water molecules to generate carbonic acid, which subsequently ionizes to form carbonate and bicarbonate ions. Step 1. CO, atmospheric equals CO, dissolved. Step 2. CO, dissolved tilde. Step 3, H. Cycle of nitrogen. It is challenging to introduce nitrogen into the living world. Given this molecule makes up around 78% of the atmosphere, plants and phytoplankton are not able to absorb nitrogen from it, which exists as firmly linked, triple covalent N. Free-living and symbiotic bacteria introduce nitrogen into the living world by nitrogen fixating it into their macromolecules, conversion of N. Most aquatic environments with sunlight support cyanobacteria, which are essential for nitrogen fixation. Cyanobacteria have the ability to fix nitrogen from inorganic sources. Legumes, including peas, beans, and peanuts, rely on rhizobium bacteria, which coexist symbiotically in their root nodules and supply them with the organic nitrogen they require. Azotobacter is one of many free-living bacteria that fix nitrogen. Many ecosystem activities, including primary production and breakdown, are constrained by the supply of nitrogen. Organic nitrogen is particularly crucial to the study of ecosystem dynamics. In terrestrial systems, this process takes place in 3 stages: ammonification, nitrification, and denitrification. The first step in the ammonification process is when specific bacteria and fungi transform nitrogenous waste from live or dead animals into ammonium, NH**. Second, nitrifying bacteria like Nitrosomonas use nitrification to change the ammonium into nitrites, NO. Similar organisms then transform nitrites into nitrates, NO. Thirdly, the nitrates are transformed into nitrogen gas by the process of denitrification, which is carried out by bacteria like Pseudomonas and Clostridium. This step enables the nitrates to once again enter the atmosphere. The 2 main ways that human activity can release nitrogen into the environment are when fossil fuels are burned, which results in the release of various nitrogen oxides, and when artificial fertilizers are used in agriculture, which cause surface runoff to carry the fertilizers into lakes, streams, and rivers. Numerous effects of atmospheric nitrogen on Earth's ecosystems are linked to it, including the creation of acid rain as nitric acid, HNO, and greenhouse gases as nitrous oxide, NO, which may contribute to climate change. Freshwater and saltwater eutrophication, a process where nutrient runoff causes the excess development of microorganisms, depletes dissolved oxygen levels, and kills ecosystem species, is one of the main effects of fertilizer runoff. Similar activities take place in the marine nitrogen cycle, where marine microorganisms carry out the ammonification, nitrification, and denitrification processes. In geologic time, the uplift of the Earth's surface could have transported some of this nitrogen sediment from the ocean floor to land, where it must have been absorbed into the rocks on land. A research revealed that this mechanism may actually be significant and should be considered in any analysis of the global nitrogen cycle, despite the fact that it has generally been viewed as less important than nitrogen. Fixing the atmosphere. Morford, Holton, and Dahlgren, 2011. Atmospheric nitrogen, N. Quidge. And anaerobic. Aria and fungi, I. Crates, NO. Figure 10.24 Nitrogen cycle. The cycle of phosphorus. Phosphorus is a nutrient that is necessary for all life processes. It's a key component of phospholipids and nucleic acids, and it also contributes to the structural elements of our bones, calcium phosphate. In aquatic environments, phosphorus is frequently the limiting nutrient required for growth. In nature, phosphorus is found as the phosphate ion, PO**. In addition to phosphate runoff brought on by human activity, phosphates are also released into rivers, lakes, and the ocean. Naturally, as they are weathered from phosphate-containing rock, the ocean gave birth to this rock. The bodies of ocean animals and their excretions are the main sources of phosphate containing ocean sediments, volcanic ash, aerosols, and mineral dust, however, may potentially be important phosphate sources in isolated areas. As parts of the Earth's surface are raised during geologic time, this silt is subsequently transported to land. Additionally, phosphate that is dissolved in the water and marine ecosystems exchange phosphorus back and forth. The average phosphate ion has an oceanic residence time of between 20,000 and 100,000 years, and it moves from the ocean to the land and through the soil at a very sluggish rate. The phosphorus cycle. Crop. Harvest. Animal S manures. And biosolids, plant residues. Organic phosphorus, microbial. Plant residues, humus. The excessive rain of microbes and the depletion of dissolved oxygen caused by excess phosphorus and nitrogen that enters these ecosystems from fertilizer runoff and sewage causes the mortality of numerous ecosystem species, including shellfish and fish. Dead zones in lakes and at the mouths of many significant rivers are the result of this phenomenon. L input to soil, L loss from soil. Atmospheric deposition, dust. Mineral fertilizers. Apatile, erosion. Mineral surfaces, days, FeN. Al oxides, carbonates. Secondary compounds. CAP, FEP, MNP, AIP. Figure 10.25 Phosphate cycle. The cycle of sulfur. Sulfur is a component that contributes to the creation of disulfide bonds inside proteins, which aid in determining the 3D folding patterns of proteins and subsequently their functions. It is a component of the amino acid cysteine. Sulfur dioxide, SO, the atmospheric version of which is produced by the breakdown of organic molecules, enters the atmosphere through 3 different processes: volcanic eruptions, geothermal vents, and human combustion of fossil fuels. Precipitation, direct fallout from the sky, rock. Weather and geothermal vents are the 4 main processes through which sulfur is deposited on land. Sulfur is present in the environment as sulfur dioxide, SO, and dissolves as weak sulfuric acid as rain falls through the atmosphere, H, SO. In a process known as fallout, sulfur can also fall directly from the atmosphere. Additionally, sulfur is released into the soil during the weathering of sulfur-containing rocks. These rocks are made of ocean sediments that have been geologically elevated and transported to land. These soil sulfates, SO, can then be used by terrestrial ecosystems, which release the sulfur back into the atmosphere as hydrogen sulfide, H, S gas once the organisms die and decompose. The global sulfur cycle's equilibrium has been significantly altered by human activity. Greater amounts of hydrogen sulfide gas are released into the atmosphere when huge amounts of fossil fuels, particularly coal, are burned. Acid rain is a phenomena that is created when raindrops pass through this gas. Acid rain is corrosive rain that harms aquatic habitats by converting rainfall into weak sulfuric acid as it falls to the ground through sulfur dioxide gas. Acid rain, SOOL, sunlight, harms both the natural and built environments. Lowers the pH of lakes, which kills a lot of the local fauna, and causes buildings to deteriorate chemically. For instance, acid rain has over time caused considerable harm to numerous marble monuments, including the Lincoln Memorial in Washington, D.C. These illustrations highlight the extensive impacts of human activity on the ecosystem as well as the obstacles we still face today. Sulfur cycle. Ln H equals 1 LAN. Figure 10.25. Biogeographic regions of the Earth. Research known as biogeography studies tries to explain how populations, species, and ecological communities are distributed over Earth. There are only a limited number of potential responses to the query of why a species is or is not present in a certain region. Explaining species distributions may appear to be a straightforward task. Reduced sulfur sum equals ULT. Sulfur cycle. When a species is present in a given location, it either evolved there or it evolved elsewhere and diffused to the area. When a species is absent from the area, it either evolved elsewhere and never distributed to the area, or it was previously present but no longer resides there. Finding the correct response involves knowledge of the evolutionary histories of the various species, which can be learned from fossils, and understanding their phylogenetic relationships. It also needs details on how the geology of Earth changes the species evolved, including information on continental drift, glacial advances and retreats, changes in sea level, and mountain construction. Such geological knowledge can help us to determine whether species dispersed and colonized new places from a remote area of origin or whether they evolved where they are currently located. It is possible to divide the Earth into several significant biogeographic zones due to the differences in the biotas of the continents. Based on the taxonomic similarity of the creatures that inhabit them, biogeographic areas are defined where species compositions drastically shift over short distances. Biogeographic regions' borders are drawn. An endemic species is one that can only be found in a certain area. Due to the significant immigration restrictions imposed by water barriers, isolated islands generally contain diverse endemic biotas. Allopatric speciation frequently creates distinct species and groups if the islands are big enough or are a part of an archipelago. For instance, Madagascar, a sizable island off the eastern coast of Africa, is almost entirely indigenous tracheophytes and vertebrates. Madagascar might be considered a biogeographic region in and of itself, but islands are regarded differently from continents because dozens of them would meet the criteria based on the distinctiveness of their biotas. The majority of species are restricted to just one biogeographic area. The greatest egret, osprey, peregrine falcon, and barn owl are a few more species that may be found in every continent but not Antarctica. Homo sapiens is most likely the most common species today. Terrain-based biomes. Ecologists distinguish between biogeographic areas and biomes, which are distinct ecosystem types depending on the composition of their main vegetation. Wherever on Earth a biome is situated, its vegetation will look similar. Despite their visual similarity, the plant species that make up these communities may not be closely linked in terms of evolution. The biome comprises numerous species in all other taxonomic groupings, but they are named for and distinguished from one another by their distinctive flora, which is occasionally accompanied by a description of their location or climate. High latitudes and high mountains are where you can find tundra. In all latitudes, the Arctic and high mountains are home to the tundra biome. Permafrost, or permanently frozen soil, lies beneath the short perennial plants that make the Arctic tundra vegetation. During the brief summers, when the sun shines continuously, the top few millimeters of soil melt. Poland Arctic tundra is very wet despite minimal precipitation because water cannot drain through the permafrost. Plants only grow for a few months a year. The majority of Arctic tundra animals either migrate to the region just during the summer or spend most of the year dormant. Boreal woodland can be found in temperate zone mountains at lower elevations and equatorward tundra. Boreal forest summers are brief, and winters are lengthy and extremely cold, although often warm. Evergreen trees benefit from the short summers since they can begin photosynthesizing as soon as the spring temperatures rise. Evergreen coniferous gymnosperms make up the majority of the boreal forests in the northern hemisphere. The southern beech, Nothofagus, some of which are evergreen, is the most common tree in the southern hemisphere. In both hemispheres, evergreen forests can be found growing along the west coastlines of continents at middle to high latitudes, when the summers are cool and dry and the winters are moderate but very wet. The tallest trees on Earth can be found in these forests. There are not many tree species in boreal woods. The dominating creatures, like moose and hares, consume leaves. Rodents and birds are supported by the seeds in pine cones. Eastern North America, Eastern Asia, and Europe are all home to the temperate deciduous woodland biome. In these areas, The temperature varies greatly from summer to winter. Throughout the year, precipitation is distributed very evenly. The majority of the trees in these woods are deciduous, which shed their leaves in the chilly winters and develop leaves that quickly photosynthesize in the hot, humid summers. Compared to boreal forests, there are many other types of trees here. The temperate forests with the most species diversity can be found in eastern China, eastern Japan, and the southern Appalachian Mountains of the United States, regions that did not experience glaciation during the Pleistocene. There are many genera of both animals and plants that are present in all three of the distinct deciduous forest biomes. There are various locations around the world where the temperate grassland biome can be found, and they are all generally dry for most of the year. The majority of grasslands, including the Great Plains of North America, the Veldt, South Africa, and the Pampas of Argentina, experience hot summers and comparatively mild winters. This biome's majority has been turned over to agriculture. California grasslands receive most of their precipitation during winters, whilst other grasslands receive the majority of their precipitation during summers. Great Plains, Russian steppe. All grassland vegetation has a straightforward structural makeup. It is abundant in perennial grass, sedge, and forb species. When forbs are in bloom, grasslands can explode with fire. Plants in grasslands are suited to fire and grazing. After being burned or grazed, they quickly regenerate because they store a large portion of their energy underground. High and arid deserts are cold. The desert biome can be found in dry areas at mid-latitudes and higher, particularly in the interiors of big continents under the rain shadows of mountain ranges. Temperature fluctuations throughout the seasons are several low-growing shrub species predominate. In cold deserts, in the winter, the soil's top layers are replenished with moisture, and in the spring, plant development is at its peak. The yearly primary yield is poor because the soils dry out quickly in the spring. Although most taxonomic groupings of species are somewhat underrepresented in cold deserts, this biome has a rich fauna of seed-eating birds, ants, and rodents because of its plant's propensity to generate vast quantities of seeds. Around 30 degrees latitude, hot deserts begin to appear. The belts that make the hot desert biome are located at latitudes 30 degrees north and 30 degrees south, and are where air falls, heats, and gathers moisture. The majority of the rain that falls on hot deserts occurs in the summer. However, storms that form over mid-latitude oceans can also bring rain in the winter. The heart of Australia and the African Sahara Desert are the driest huge areas, where summer and winter rains seldom ever fall. Hot deserts feature richer and more architecturally diversified vegetation than cold deserts, with the exception of these driest areas. In some hot deserts, succulent plants that contain a lot of water and their expanded stems are noticeable. Annual plants produce a large number of seeds that sprout when it rains. Animals frequently disperse fruit and perform pollination. The abundance of rodents, termites, and ants is frequently astounding, and the species diversity and abundance of lizards and snakes are usual. Chaparral has a dry, pleasant climate at moderate latitudes, about 30 degrees when mild. Ocean waters flow offshore. The chaparral biome can be found on the western flanks of continents. This biome experiences chilly, rainy winters and warm, dry summers. The Mediterranean region of Europe, coastal California, central Chile, extreme southern Africa, and southwestern Australia all have similar climates. Low-growing shrubs and trees with robust, evergreen leaves make the majority of the vegetation in chaparral. In early spring, when insects are active and birds are laying their eggs, shrubs perform the majority of their growth and photosynthesis. Annual plants are widespread and produce a large number of seeds that land on the ground. As a result, this biome is home to enormous number of tiny rodents, the majority of which deposit their seeds in underground burrows. The flora in the chaparral has evolved spontaneously to withstand recurrent fires. In late fall, when a significant number of migrating birds arrive from the north, many bushes in chaparral in the northern hemisphere yield fruits that are spread by birds. Savannas and thorn forests have comparable climates. On the equatorial sides of scorching deserts, thorn woodlands grow. Semi-arid conditions prevail. Winter months may see little or no precipitation, but summer months may see significant amounts. There are numerous plants in thorn forests that resemble those in scorching deserts. Small trees and prickly shrubs, many of which lose their leaves during the long, dry winter, make the majority of the vegetation. Acacia species are widespread in thorn woodlands all over the world. Africa, South America, and Australia all feature sizable areas of savannas, which are vast fields of grasses and grass-like plants with a few scattered trees. The greatest savannas are found in Central and Eastern Africa, where the biome is home to numerous large carnivores that feed on grazing and browsing mammals, as well as a significant population of these animals themselves. The savannas are kept up by the browsers and grazers. Savanna vegetation usually turns back into a dense thorn forest if it is not grazed, browsed, or burned. In heated lowlands, tropical deciduous woods can be found. Tropical deciduous woods replace thorn forests as the length of the rainy season lengthens toward the equator. These forests are far more diverse in terms of plant and animal diversity than thorn forests, with larger trees and less succulent plants. During the protracted, hot dry season, most trees lose their leaves, with the exception of those that are found around rivers. The majority of their species are pollinated by animals, and many of them flower while they are without leaves. The hot, wet season is when biological activity is at its peak, because they are less nutrient-leached than the soils of wetter regions. The soils of the tropical deciduous biome are some of the best soils in the tropics for agriculture. Due to this, the majority of tropical deciduous woods have been destroyed to make room for farming and cattle grazing. In various continents, restoration activities are in progress. Tropical evergreen woods have a join with upper line. Equatorial places where annual total precipitation surpasses 250 centimeters and the dry season does not last more than 2 or 3 months, tropical evergreen forests can be found. They have the highest diversity of plant and animal species of any biome, with up to 500 different tree species found in every square kilometer. Tropical evergreen forests have the highest total productivity of all biological communities in addition to having an enormous diversity of species. The majority of mineral nutrients are, however, bound by the flora. Normally, soils cannot support agriculture without extensive fertilizer applications. Tropical mountain slopes have shorter trees than tropical lowlands do. There are more epiphytes, plants that grow on other plants and get their nutrients and hydration from the air and water instead of soil, and their leaves are smaller. The island biogeography examines patterns in species distribution on islands is influenced by ecological and evolutionary processes connected to island characteristics like isolation and area. A MacArthur-Wilson hypothesis of island biogeography: the species diversity of an island's biota is determined by two processes, immigration and extinction. The immigration rate declines and the extinction rate rises as the number of species on the island rises, while the extinction rate falls with island area. The immigration rate decreases with isolation, distance effect, area effect. A balance between species diversity and immigration and extinction rates is reached. Species turnover occurs when an island goes extinct and is replaced by the arrival of new species. One of the ideas that have had the most influence on contemporary biogeography, ecology, and evolutionary biology is the equilibrium theory of island biogeography. Additionally, it has had a significant impact on conservation biology, especially with efforts to create a theoretical framework for the creation of nature reserves. The equilibrium theory of island biogeography should not be blindly applied to nature. Conservation, as several components of that theory are still unproven. A theory of island biogeography's equilibrium species-area relationship. It has long been known that there is a monotonic increase in the number of species with increasing island area or sampling area of terrestrial communities. In many instances, a power function with the following shape has been demonstrated to best suit observations among the different mathematical representations for the species-area relationship. S equals log or cas1, S equals z log. S is species diversity, the number of species, also known as species. Richness, A is area, and Sen and Z are positive constants where S is the number of species. Theoretical value of 0.263 and typically varies between 0.8 and 0.35, while C typically indicates the impact of geographic variation on species diversity. The usual method for estimating values for these constants is linear regression. Although the aforementioned equation has been used frequently, it has limited ability to explain or predict. As a result, to explain the species-area relationship, Williams' 1964 habitat diversity hypothesis, Connor and McCoy's 1979 passive sampling hypothesis, and Munro's 1948, 1953, Preston's 1962, and MacArthur and Wilson's 1948, 1962 dynamic equilibrium hypothesis. 1963, 1960. The species-area link, according to diversity hypothesis, is the outcome of a positive correlation between area and habitat diversity as well as between habitat diversity and species diversity. Passive sampling hypothesis simply views isolated taxonomic groups as samples or subsets of broader populations. Therefore, sample size and intensity both affect species diversity. Coleman, et al., 1982, offered a related theory, known as the random placement hypothesis, that goes along these lines. The article's focus, the dynamic equilibrium hypothesis, has had the most impact on island biogeography and ecology. Area impacts the observation that as island acreage grows, species extinction rates decrease. Effective distance, the observation that as an island is further from its continental source of colonizing species, the rate of species immigration declines. Evolutionary curve is the line connecting the number of species already existing on an island to the rate of species extinction. Migration rate is the line connecting the pace of species immigration to the total number of species on an island. Biogeography of islands. The end of patterns in species distribution on islands is influenced by ecological and evolutionary processes connected to island features such locality and isolation. When conspecific immigrants arrive, existing populations receive demographic support and their genetic diversity is increased, which reduces the rate of species extinction. Curve of species area is the line connecting species diversity to island size. Variety includes either the total amount of the number of species, species richness, or includes both the species count and relative abundance of them. The biosphere, or the web of life that risen is dependent upon the inorganic spheres, is an essential component of man's ability to live a long, healthy life and necessary for the existence and functional state of human species. Our life is made feasible and delightful by the diversity of living things that makes the biological world. Walter and Rosen, 1985. First used the term biodiversity, which is the shorthand for biological diversity. Nearly 4 billion years ago, life first appeared on Earth, and it took nature more than 1 billion years to create the vast and intricate variety of life that exists today. According to scientists, there are between 10 and 80 million different species on the planet. Wilson, 1988, of which 1.4 million have been officially recorded. However, we're rapidly losing this history that dates back millions of years. The entire living world will undoubtedly suffer serious effects from the decline in life's species diversity. Studying the many types of life on Earth and the factors that contribute to their extinction has become of utmost importance. The overall variety of life on our planet is known as biodiversity. Biodiversity is the sum of all the different types of bacteria, plants, and animals that are present in a system, including their numerous races, variations, and species. The term, biodiversity, is currently used often by a wide range of people, including the general public, environmental organizations, environmentalists, business people, and economists. Therefore, it is crucial to understand the definition of biodiversity, which is acknowledged as a distinct science with its own guiding principles. The following are some key definitions of biodiversity. 1. Biodiversity is the diversity of life in all of its manifestations, scales, and configurations.